Welcome once again to Masterclass in Biology. Today we will be discussing MVQs on microbes in human welfare. So normally there are three questions from this chapter. So it may be a chapter that is not very big in content, but it is important for the NEET entrance examination and one of the chapters where examiners frequently ask you questions. Now, the most valuable questions are most likely to appear in the NEET entrance examination and therefore are based on the text matter of your NCERT textbook. This is what we have told you time and again. So now is the correct time to keep the NCERT handy with you and look at the NCERT text matter as and when I am saying something about the text matter, just make a note and look the text matter of your NCERT so that it becomes a part of your memory. So first thing first, if you look at this, microbes in human welfare, you must understand that NCERT includes everything that is given in biological classification chapter as microbes. So viruses, viroids, prions, look at the NCERT, they are included in microbes. Now traditional biotechnology is use of these microbes, the biological processes such as fermentation, to get services and products that are used for the welfare of the mankind. So this thing is what is called as traditional biotechnology. The modern biotechnology is manipulation of the genetic material, what is called as the genetic engineering, and then making the products which is called as the bioprocess engineering or biochemical engineering, and together they are called as the modern biotechnology, sometimes called as recombinant DNA technology as well. So we are looking at traditional biotechnology here. Now, the first question, see, three questions can come from this chapter, but they are very straightforward. So question number one would be, in the household products, they are asking you about the lactic acid bacillus. During fermentation produces B12. So if you are fermenting a curd or the curdling of the milk is actually achieved with the help of this microbe, it adds to the nutritional value and it adds vitamin B12, which is very important, at least for the vegetarians. Number two, because it can survive in the acidic environment, it gives a tough competition to Helicobacter pylori type of bacteria that are very common causes of peptic ulcer disease, and therefore can protect the human being from this particular disorder. This is about lactic acid bacillus. Then obviously you know about Saccharomyces cerevisiae. This is a very important question. Baker's yeast or brewer's yeast, you know that the fermentation is alcohol fermentation. So ethyl alcohol or ethanol or carbon dioxide is produced, and in bread, say for example, the carbon dioxide gets trapped in the dough, raises the dough. The process is called as proving. And similarly in fermentation there is ethanol fermentation, and in the ethanol fermentation, remember your class 11th chapter where it says that the natural fermented alcoholic beverages do not have more than 13-14% of alcohol content, because this percentage becomes toxic to the microbe itself, and therefore the fermented alcoholic beverages, say for example like whiskey, brandy, rum, etc., where the content may be around 40% or even more. How is it possible? This is possible with distillation. So a very common question is that what are the alcoholic beverages which are produced with distillation, which are produced without distillation? Without distillation would be wine and beer. This is an important question. Now they have given you a palm sap can be fermented to produce a traditional alcoholic drink in South India, which is actually called as toddy. And then there is the dough that is produced for, or batter that is actually produced for South Indian delicacies like dosa and idli. They are actually fermented with the help of bacteria. And then finally there is an important question on cheese. So cheese is actually, cheese can be unripened, what is called as cottage cheese. That means it has not been treated with microbes, but the ripening of the cheese is achieved with the help of microbes. Mind the word ripening. So ripening adds a particular texture and a flavor to the cheese, and the microbes are used here, and the microbes can be bacteria, can be fungi very frequently, and can be both. So say for example, the Roquefort cheese is ripened with the help of mold which is called as Penicillium roqueforti, and camembert is ripened with the help of Penicillium camemberti. And there is an important question about Swiss cheese, that what is the reason for large holes in Swiss cheese? The answer is that Propionibacterium shermani actually produces large amount of carbon dioxide during fermentation, that is responsible for the large hole size in the Swiss cheese. So what I am doing is that I am bringing the NCERT. This is what is microbes in household products. Similarly, in industrial products, we have talked about the fermented beverages, and now we come to antibiotics. Now, in antibiotics, please remember there is a question that what the antibiotic is antibios, which means against life. Now, this is in context for the microbes. So suppose a bacteria enters a human body, you take antibiotic, you want to kill this bacteria. So this is actually antibiotic for the pathogen, but it is actually should be regarded as a probiotic for the human beings. Generally, the term probiotic is used in another concept where microbes are used as nutritional supplements. Lactic acid bacteria, Saccharomyces boulardii are two very, very important microbes that take care of the gut health. So this is very important. Then they talk about penicillium or penicillin antibiotic, which was discovered serendipitously by Alexander Fleming, and Alexander Fleming was actually raising a bacterial culture, and when he saw that the culture was contaminated with the blue mold, then the bacteria were unable to grow, and he could understand that this mold is producing something that is actually killing the bacteria. That led to the discovery of penicillin. The species used was Penicillium notatum. Now, normally it is Penicillium chrysogenum because the yield is higher, but a simple discovery was not enough for commercialization or converting the molecule into a drug that can be used by the patients. And then there comes the role of Flore and Chain. They were able to find a way to make an actual medicine out of it, and how it can be given to the patients. Phenoxyestic acid is a molecule that stabilizes the structure of penicillin. It is grown in 2% mol agar. So all the modalities were actually worked out by Flore and Chain, and then your NCERT says that Fleming, Flore and Chain got shared the Nobel Prize. And then NCERT says that do you know that there is an in-text question? Let me show you the in-text question here. There is a, can you name some other antibiotics and find out their sources? So please remember Streptomyces. So it is a bacterium. So we are talking about Streptomyces, and please remember the name of Streptomyces. It is a source of many antibiotic, and as you can see, one would be streptomycin, the other would be tetracycline, the third would be erythromycin, fourth would be greasephulvin. These are all the source bacterium belongs to the genus Streptomyces. Now, no need of right now going into the various species that are responsible for individual antibiotic productions. And I will strongly advise you to look at the common name of whooping cough. So whooping cough is called as Kali Khansi, diphtheria is called as Galgotu, and leprosy is called as Kushtrog. And plague actually, let me tell you, is called as Mahamari, or is known as Mahamari as well. So I do not know, they can make a matching question here that okay, tell us that which disease is actually known by what name in common parlance. So I hope this thing is now absolutely clear. Now, chemical enzymes and bioactive molecule, I have asked many times about Aspergillus niger or Aspergillus niger. So this is a fungus, and this is very important question asked many times in entrance examination. This is used for the commercial production of citric acid, and it is also the source of an enzyme which is called as glucoamylase. It is used in the production of high corn fructose syrup, and it is also the source of an enzyme which is called as glucose oxidase, that is used in the glucometers that you see being used to actually check the blood sugar level. So the organic acids, obviously organic production of organic acids with the help of microbes is one. The second would be the clearing of the fruit juices. So clearing of fruit juices, and they are with the help of enzyme. More commonly it would be pectinase. It will dissolve the middle lamella, and obviously you are using certain proteases as well. So these similarly there is an enzyme that is used in meat tenderization. So meat tenderization is papain. So this is not given in NCERT. So let me tell you that meat is tenderized with the help of papain. The third one is that biological detergents. So biological detergents, and specifically NCERT is talking about lipases and detergents, and NCERT is talking about lipases, and they are used in removal of oil stains, etc. So similarly there are some enzymes that are derived from microbes that are used to actually recover silver from the photographic films. So again proteases. So the silver is impregnated in the photographic film when the film is exposed. Now nobody is using photographic films, but just to give you an idea that you can actually regenerate silver with the help of the proteases as well. So this is where there are some of these uses of the microbes are given, and this is where I have added one or two, and please try to remember them. And then we are looking at Streptococcus pyogenes, and this microbe produces a thrombolytic. So this is producing a thrombolytic. This we have discussed in body fluids and circulation as well. So this is a thrombolytic, and it is used to dissolve intravascular clots. Say for example in the cases of coronary artery disease, because it is of a bacterial origin, it cannot be used again because of antigenicity, and therefore appropriately your NCERT calls it as a clot buster. So this is an important molecule that is derived from the microbes. Now please remember this. There are better substitutes than streptokinase. Now we have urokinase and a recombinant DNA product which is actually called as tissue plasminogen activator. So this is about the clot buster. And then the selective immunosuppressant. So selective immunosuppressant. So this is selective, and this is called as cyclosporin A, and the NCERT says that it is derived from Trichoderma. So this is derived from Trichoderma polysporum. So this is the fungus that is the source of cyclosporin A. Now we are following NCERT, and many people say that the fungus is actually something else, but right now is not the time. We have to follow NCERT. It is selectively immunosuppressant. That means selectively suppresses the T cells, and therefore has a very important role in organ transplant surgeries where the graft rejection is avoided with the help of these immunosuppressant drugs. So this is something very important. And then there is another molecule that is given which is called as statins. So statins are the products of Chinese red vent yeast, and the name of the yeast is Monascus purpureus, and this Monascus purpureus actually produces some molecules which are called as monocolins, and one of them would be called as statin, and this statin is a competitive inhibitor. It is a competitive inhibitor of cholesterol synthesizing enzyme in the biosynthesis of the cholesterol, and therefore is used as a blood cholesterol lowering agent. As you are aware that hypercholesterolemia is actually a risk factor for coronary artery disease and cerebrovascular accidents. So it becomes a very, very important molecule from which you get a product. So I hope this thing is now absolutely clear. Make sure that you know this absolutely clearly, because this is where a question can easily be formed. Then about the sewage treatment, what exactly is the question? Sewage treatment, what exactly is a sewage? So I'm not going into this. You know about the domestic and municipal waste which is called as sewage, what it is rich in. It is rich in organic matter. It can contain the microbes like E. coli and Salmonella typhi, and there is a primary there is a primary treatment. In the primary treatment, first you do the filtration, then you do the sedimentation, you get what is called as primary sludge, which is actually discarded, and then the supernatant is taken to the aerobic tanks, aeration tanks for secondary treatment, and in the secondary treatment the microbes are used. This is the biological part of the sewage treatment. I am just revising the question, and you measure the biochemical oxygen demand. Higher the BOD, more organic matter is present, lower will be the dissolved oxygen content, and therefore you will have to aerate the system so that the growth of the aerobic bacteria already present or added as an inoculum into the primary effluent actually increases. They form flocks. They can be associated with certain fungal elements as well, and they form flocks. This is what you want. They will consume the organic matter, and this organic matter will be consumed, so the BOD gets reduced and the dissolved oxygen content is increased, and we are not making water drinkable. We are just making water so much pollution free that it can be added to the natural water bodies, say for example rivers. And what happens is that after this again you allow sedimentation, and this time what you get is an activated sludge. So what you get is an activated sludge, and the supernatant is actually passed on to the water bodies like rivers. So this is now sufficient enough pure to be added into the water bodies. The activated sludge is taken for anaerobic sludge digester. And please remember that some part is actually preserved to be used as an inoculum for the incoming batches in the primary treatment, but majority of them would be taken into the anaerobic sludge digesters. And what happens here is that these anaerobic bacteria, they digest this biomass and they convert this biomass into gases like methane and carbon dioxide, and that would be the production of biogas. So these are the questions that they can ask you from sewage treatment. Please remember, very important part is secondary treatment or biological treatment. Now again, we have already discussed about the biogas. So biogas, they will ask you what is the composition. The predominant thing is methane, and it can contain carbon dioxide in a significant amount and traces of hydrogen sulfide. There are other gases as well, but we are limiting ourselves to NCERT. You require a biomass that will be converted into biogas by bacteria like methanogens. So you require a biomass and you require methanogen. Obviously, the two questions possible. One would be that where do you get that methanogens from? Say for example, it is present in the rumen of the ruminants. This is a place. And similarly, you know about the cow dung, which is called as gobar in North India. And this is where the methanogens are present, and that is why it can actually be used as a biomass for production of biogas. And such biogas that is being produced in rural India is appropriately called as the gobar gas. Obviously, the constitution of the biogas will vary according to the biomass that is being used, and because the details of these steps of biogas production is not required here. And please try to remember, it is very good for rural sector where this biomass is easily available, and the Indian Agriculture Research Institute and KVIC have been instrumental in setting up the biogas plants in rural India. These are the questions that are likely to be asked from this particular thing. And then we are talking about biocontrol agent. Now this is something very important. Now try to understand, if there is a pest, and this pest actually, and you use a chemical pesticide. Now this chemical pesticide may be very effective but is non-specific, and that means it can damage, it can actually cause hazard to the useful insects that may be present in the ecosystem. They may be very important pollinator, they may be rendering other services, and they can be toxic to the human beings as well. So therefore, the need in organic farming is is to actually somehow do not try to use the chemical fertilizers, and rather than they control the pest population with the help of living organism, and this is what is called as biocontrol. So organic farmers understand that more biodiversity, more stability and more productivity of the ecosystem, and they can understand that it is virtually impossible to eradicate a pest from a particular place, and therefore the pest populations are kept at a very low yet manageable level by these type of biocontrol agents. So this is where especially in the ecologically sensitive area, this is where organic farming is very, very important. You also know, we will also see that they also use biofertilizers. So let us look at the organic farming. So what organic farmer is doing is not relying on the chemicals and is trying to use the waste, for example, from a process as a resource for the another process within the system that he has actually created. The reliance is on the living organisms. So these living organisms are used as biocontrol agents. They are also used as a biofertilizer, and this is the recycling that the organic farmer is using. This is very, very important in integrated pest management, especially in the ecologically sensitive area. So now you can understand both topics, that what exactly is happening here. At one end we are using the biocontrol agent, at the other end we are using biofertilizer. And when you look at the biocontrol agents, then look at the ladybird controls, aphids, and similarly dragonfly controls, mosquitoes. We will talk a lot about Bacillus thuringiensis as biotechnology. So now I am not going to talk a lot about Bacillus thuringiensis, just reminding you that it can form spores, and these spores can be used in the form of sprays to protect plant against some insect, arthropod pathogens or pests. So I hope this thing is now absolutely clear. And Bacillus thuringiensis has been used as a bioinsecticide, as I told you that we will discuss it a lot in biotechnology. Now, for the control of many pathogens in the rhizosphere, so around the root of the plant there are many pathogens that include fungi. There is a fungus that is used as a biocontrol agent, and this fungus is called as Trichoderma harzianum. So this can control not only fungus but many other pathogens that are present in the rhizosphere. Rhizosphere will be the area in the soil where the plant roots are actually present, and they are very, very important in preventing the diseases in plants. And then your NCERT talks about the baculoviruses. Now baculoviruses belong to genus Nucleopolyhedrovirus, and they are narrow spectrum and species specific insecticides, and as I told you, very, very critical in the integrated pest management. And after this biocontrol, let us look at the next thing, which is natural corollary, which is as biofertilizer. Again, the NCERT is talking about organic farming. I have told you that please make a list of all the diazotrophs. All the diazotroph must be a fact sheet. There is one question that is virtually guaranteed in the NEET entrance examination, which are free living, which are symbiotic, which are free living as well as symbiotic, and which are aerobic, which are anaerobic. Make a column wise list and try to remember which are nitrifying bacteria, which are denitrifying bacteria. Collect the information from the mineral and nutrition chapter as well. Make a comprehensive list, and out of this, what actually fixes nitrogen, say for example in cycas, or say for example what fixes atmospheric nitrogen in leguminous plant roots, and what is the role of Frankia, how do they protect their nitrogenase examination. And nitrogen cycle is something along with the name of these microbes that you can be assured that there would be at least one question in the NEET entrance examination you are going to face. So now is the time. If you have not done so, now please make a comprehensive list list and try to remember the names and the importance of the cyanobacteria, especially Azolla anabina relationship, and the importance for the paddy cultivation of this symbiotic relationship. Similarly, about the glomus, the mycorrhiza, and the role of mycorrhiza in making water available to the plant, increasing the surface area, fighting the pathogens, environmental conditions when they get perturbed, and therefore the plants are made resistant to them, say for example drought, etc., and the availability of minerals like phosphorus. And this is the important role of mycorrhiza. So I hope you have understood the text matter given in NCERT. Rest assured there can be 2 or 3 questions from this chapter in your NEET entrance examination. Thank you so much.